Okay, everyone. Well, thank you so much for coming. It's my absolute pleasure to introduce Keilani, a graduate student in the Hilo Tropical Conservation Biology and Environmental Science program. She's going to be defending her master's thesis today. It didn't start here. Keilani was born and raised in Waimanalo, Oahu. She's always been fond of the beach and the outdoors, which led her eventually to doing a geology degree in California at Occidental College. She got really into rocks and mountains and exploring the coastline, started working with geology groups where she began remote sensing, trying to understand erosion on the coastlines, which I think kind of instilled a little bit of passion to go back to Hawaii and look at coastal issues and water quality, where she worked studying water quality actually for a few years after she graduated, realized she had more to do and wanted to come back to school, expand her expertise, where she joined TCBES and started working with us in the Mega Lab. And she shot out of a cannon when she got here and started doing 3D surveys all over the island with great big plans. And just as fast as the progress started, things turned down with the COVID pandemic, some of you may be aware of. And so she had to pivot. And I have to say, she did a phenomenal job sort of restructuring her project to deal with that setback, and reached out and collaborated with several folks at NOAA, including Tom Oliver and Dr. Tom Oliver and Dr. Courtney Couch, and created a really unique way of looking at how coral reefs are changing through time in the Northwestern Hawaiian Islands. So she was able to take the skills she had learned and expand into an area where there were some existing data sets where she could really sort of change how we approach understanding shifts in these communities through time. So it's been a pleasure working with Keilani. We're sad to see her go, but we're very excited for all of her accomplishments. And so with that, I will introduce her project, Examining Patterns in Coral Demographic Events in the Papahānaumokuākea Marine National Monument. Take it away, Keilani.
Thanks so much for that awesome introduction, John. So yeah, I'm Nola Keilani Bonis-Ericksen, and my master's research is examining patterns in coral demographic events in Papahānaumokuākea Marine National Monument. And I worked on this project with my advisor, John, and my committee members, Grady and Jolene, and they really pushed me to be the best researcher, scientist, and writer I could possibly be. And for that, I'm so grateful. So thank you. And I also worked on this with NSF-funded Ikebai students and had support from NOAA researchers, including Dr. Tom Oliver. And I really couldn't have done this without the support of all my friends and family. So if you guys are here tuning in, thank you. And with that, I'll begin. So coral reefs are some of the most productive ecosystems in the world, and they provide dynamic habitats capable of supporting high levels of diversity and abundance of reef organisms. They provide services through fisheries, tourism, recreation, and coastal protection, and each year contribute over $3.4 billion into the US economy. Coral reefs are affected by both global and local stressors. So some examples of global stressors are climate change and ocean acidification. And some examples of local stressors are pollution, overfishing, sedimentation, and coastal development. And as you can see, this background photo here is of bleached coral at Honaunau, which can happen when temperatures exceed a threshold temperature for an extended period of time. So as these stressors increase in intensity, some studies have predicted that coral reefs could be reduced by up to 90% by the middle of the century. So considering the impacts of climate change and stressors on coral reefs, studies must identify mechanisms of resiliency, stress, and mortality to inform managers and prevent degradation of reefs. So first, let's start with, what is a coral? Corals are sessile colonial organisms made up of building blocks of connected polyps. They rely on their mutualistic relationship with many microorganisms, like the photosynthetic dinoflagellate Symbiodinium, which provides nutrients that the coral needs to survive and grow. The unique relationships that corals have with these microorganisms causes them to have a complex biology in which they can have high phenotypic plasticity, meaning they can exhibit many different morphologies like these shown on the slide here. So this nature of corals to exhibit many different morphologies means that different corals respond differently to their surrounding environment. Identifying a coral in the field is easier said than done. For example, this is a really obvious postleporid coral colony. Can everybody see that? So how many corals do you think are surrounding this colony here? I ask you this so you can see that some corals are super easy to identify, just like this Postlepora meandrina here, but some are really hard, like the surrounding coral. And here are two very difficult colonies to identify. How many corals do you think are in each of these images? So I just want you to think about this for a bit while I continue. As some species age, a coral can split into many different pieces. So there's two schools of thought on identifying coral. The first school of thought says that all these pieces came from a single colony, and so they should be considered one coral. And then another school of thought says that once the coral splits apart, these are now independent entities, so they should be considered different colonies. And there's not really a consensus as to which school of thought is correct. So if you guessed on the left, this is actually one contiguous coral patch, but on the right, it splits up into 28 patches. So what no one can really decide on in the scientific community is if these 28 patches are considered part of one individual coral colony, or if they are separate. So that's why in my study, we considered each separate and identifiable area of contiguous live tissue a coral patch. And if we treat each live tissue area as a patch, then the issue of whether patches are of the same coral colony becomes irrelevant, because we're now just looking at how each patch changes through time. And patches create the coral reef community, which are a foundation for the reef ecosystem. So ultimately, the fate of a coral patch dictates the fate of the overall reef. And shout out to Dr. Tom Oliver, who came up with this new approach of using patches to understand changes in corals through time. Identifying how individual coral patches change over time is critical for predicting how coral reefs will change in response to environmental stressors. This leads me to my objectives, which is to first examine changes in coral patches like shrinking, growth, fission, fusion, mortality, and recruitment through time in Papahānaumokuākea Marine National Monument. And also to explore how coral patch characteristics like size and genus and site characteristics such as coral cover and reef complexity are associated with the likelihood of a coral patch experiencing the changes that I just mentioned above. I use data from Papahānaumokuākea Marine National Monument, which is in the northwestern Hawaiian Islands and is one of the largest protected areas in the world, encompassing multiple islands, atolls, and reefs, and they're home to many endemic organisms. This monument provides a unique natural setting to study the effects of global anthropogenic stressors on coral reefs through time, as if its reefs are pristine and largely unaffected by direct human impact. And specifically, my study used data from Pearl and Hermes, Lisiansky, and French frigate shoals, which are shown in the red boxes. Initially, I didn't exactly set out to work in the northwestern Hawaiian Islands. I was going to collect my own data all around Hawaii Island, but as we all know, COVID happened, and so this restricted my ability to conduct any sort of field work. And this is actually what motivated a collaboration with NOAA and Dr. Tom Oliver to use time series data that had already been collected by the Multi-Scale Environmental Graphical Analysis Lab, or MEGA Lab, and NOAA. So even though this data was already collected, no one had ever analyzed it using this new approach of tracking coral patches through time. And along with 11 amazing Hilo students, we were examining these coral reefs with an entirely new perspective. And just a shout out to EPSCoR Ikevai for funding this project. So many studies have focused solely on quantifying overall coral cover through time, but overall coral cover doesn't demonstrate how coral patches of different size and genus are changing and responding to environmental stressors over time. researchers have struggled to quantify and track fine-scale temporal changes because of challenges associated with high-resolution underwater mapping of coral habitats. But advancements in the fields of photogrammetry and remote sensing have enabled researchers to track responses of individual coral patches to environmental stressors. My study seeks to identify changes that occur among individual coral patches over time in the Northwestern Hawaiian Islands. So these changes are called demographic events. What are demographic events? Over time, corals can undergo fission, when a coral patch divides into two or multiple patches; fusion, when two or more patches fuse into one; mortality, when a coral patch dies; recruitment, when larva settles and attaches to the benthic substrate to survive and grow; shrinking, when a coral patch decreases in surface area; and growth, which is the increase in surface area of a patch over time. So analyzing these fine-scale changes is key to understanding how coral reefs will respond to environmental stressors in the future. This is an example of an Acroporis Etheria that we analyzed from imagery of Rapture Reef in French Frigate Shoals in 2016, shown on the left, and then 2017, which is shown on the right. As you can see, from 2016 to 2017, this Acroporis Etheria coral underwent partial mortality and decreased in surface area, which is outlined in blue here. And then we classified this as a shrinking event. And tracing thousands and thousands of coral isn't usually overly exciting, but I thought this is a cool example of coral growth that we measured and analyzed at French Frigate Shoals. So shown on the left is an Acroporis Etheria coral in 2016. And then on the right is that same coral in 2017. And the white arrows show how much the coral grew over the course of the year. These examples show how we tracked changes in individual coral patches through time using high resolution imagery, allowing us to identify these demographic events. And just for scale, this growth was about 12 centimeters. The second component of my study was to better understand what reef characteristics can influence the occurrence of coral demographic events over time. Both site and coral patch characteristics can influence the occurrence of growth, shrinking, fission, fusion, mortality, and recruitment. And in this study, site characteristics consist of live coral cover and reef complexity. Reef complexity is the 3D topography of the reef, including any holes, crevices, and formations. In this study, coral patch characteristics consist of the genus and the size of an individual coral patch. How do we extract these demographic events and explore drivers of these events? So we use coral reef survey data from Northwestern Hawaiian Islands collected by NOAA research team, including Megalab researchers. And the Megalab has been working with NOAA researchers like Randy Kosaki and Atsuko Fukunaga to repeatedly return to the same long-term monitoring sites to collect coral health and structure for motion photogrammetry surveys. We use data from a total of 15 sites, 6 in French Frigate Shoals, 6 in Lisiansky, and 3 in Pearl and Hermes. And survey data from 2 time points spanning 2016 to 2019 were used. For the 15 study sites, researchers used scuba diving to capture hundreds of overlapping images of the substrate in a lawn mowing pattern, as you can see in this figure, to acquire images that encompassed a 10 by 5 meter area. This method, called structure from motion or SFM photogrammetry, is a cost-effective, non-intrusive, and efficient way of capturing large areas of the reef in high resolution. So after Megalab researchers collected this imagery, we inputted these overlapping images into, for 15 sites, into Agisoft PhotoScan software to produce high-resolution 3D models, as shown in this video of a Lisiansky site that was made by Alex Spengler, who is a Megalab researcher. And this is just a swim by of the model that shows the resolution down to a millimeter scale. So it's pretty awesome. Thanks for the video, Alex. Now, there are two products that we export from the model that was shown in that video, an orthomosaic and a digital elevation model. The orthomosaic is a geometrically corrected mosaic from photos that have been stitched together. And it's at such a high resolution that we can zoom in and see individual coral colonies that are even less than a centimeter in width. And with that is an accompanying digital elevation model, or DEM, which is produced from the exact same model and has cells, and each of those cells have X, Y, and Z values. So when we trace features in the mosaic, we can extract X, Y, and Z information for that specific location. From the DEM, we're able to extract metrics like surface complexity, which is the 3D over 2D area of the reef. And this ended up being a predictor variable used in statistical analysis to determine whether or not demographic events are likely to happen. We also used orthomosaics to measure and determine how much live coral is in a site, what characteristics are associated with those corals, like genus and size. And then by looking at these orthomosaics through time, we can identify demographic events. For example, did these patches grow? Did they shrink? Did they die? Or did new patches appear over time? Specifically, to identify demographic events and analyze changes in individual coral patches, we first aligned orthomosaics from different time points like 2016 and 2017. So the orthomosaics overlapped and the same coral patches could be traced for each time point. And this is shown in the figure to the left. And we randomly overlaid 21-meter diameter circles, shown in yellow, on each orthomosaic and used these as subsampling areas to trace all live coral patches. We traced all live coral patches within each circle and linked the same corals from time point 1 to time point 2. And then we identified demographic events like growth, shrinking, fission, and fusion that occurred over time. We recorded if a coral patch died and if recruitment occurred. This image is of coral patches that were traced within a 1-meter diameter circle and shows just the incredible resolution that allowed us to trace even the smallest of corals. And an amazing group of MegaLab students and I traced thousands of coral patches among the 15 study sites for both time point 1 and time point 2. So throughout this presentation, you'll notice that I frequently use we instead of I, because I could not have done this without their help. We identified, measured, and compared about 2,409 coral patches over a 2 to 3 year time span across 15 sites and recorded demographic events that occurred for each patch. This shows the proportion of each demographic event out of 2,409. So the good news is the greatest proportion of coral patches underwent growth at 31% and recruitment at 24%. When we look at the proportions of demographic events by island, Pearl and Hermes had the greatest proportion of mortality and recruitment. French frigate shoals had the greatest proportion of coral patches that underwent growth. And Lisiansky had high proportions of coral recruitment and growth. Now back to my original question, how do coral patch and site characteristics influence the occurrence of coral demographic events? To answer these questions and better understand these associations, I conducted a series of statistical analyses. So to determine statistical differences in median coral cover among islands and between time points, I applied a comparative non-parametric statistical test known as the Kruskal-Wallis analysis. I ran a binomial logistic regression model to understand the likelihood of a mortality, fission, fusion, growth, or shrinking event occurring given the genus, starting patch size, reef complexity values, and coral cover surrounding a patch. I also ran linear regression models to estimate the relationship between coral recruitment rate and reef complexity. It's really important to note that this was an exploratory analysis using model selection to understand which variables influence the occurrence of a demographic event, and this was not a controlled experiment. I use the backward stepwise regression, backward stepwise regression function in RStudio to identify what combinations of genus, patch size, coral cover, and reef complexity were the most important predictors of a demographic event occurring. The backward stepwise selection is a variable selection method, and it begins with a model containing all possible variables, and then it removes the least significant variables. And the selection criteria was the Akaike information criteria. And the Akaike information criteria, or AIC, estimates the quality of the model relative to other models to determine the model of best fit. I first looked at percent coral cover at all sites to give me a better understanding of coral cover by island. So here you can see percent coral cover among islands and between years, with red representing 2016 and blue representing 2017. The Kruskal-Wallis statistical analysis revealed that median values of percent coral cover were statistically different between islands. This makes sense because you would expect different sites and different islands to have different proportions of coral cover. And this analysis also revealed no statistical differences in percent coral cover between time point 1 and time point 2. And this is really important for my study because it indicates that there were no significant events causing dramatic increases or decreases in overall percent coral cover. So this analysis will reveal the dynamics of coral patches in an overall stable reef ecosystem. I ran binomial logistic regression models to understand the likelihood of a coral patch undergoing a particular demographic event given its genus, starting size, surrounding coral cover, and the surrounding coral reef complexity. So binomial logistic regression models are used to predict the probability that binary outcomes will occur given one or more independent variables. The dependent variable has two possible outcomes, one, which the event occurs, and 0, which the event does not occur. In this study, the dependent variables were the occurrence of demographic events like growth, shrinking, fission, fusion, mortality. So for example, when using a logistic regression model to predict mortality, if a coral patch died, we assign this a value of 1, while all other events, growth, shrinking, fission, fusion, were assigned a value of 0. And for predicting growth and shrinking, if a coral underwent growth, we assign this event a value of 1. And if a coral underwent shrinking, we assign this a value of 0. The predictor or independent variables were a combination of patch size, genus, surrounding coral cover, and reef complexity. Since the coral patch data was really skewed, with some coral patches centimeters long, while other coral patches were meters long, I used the natural log of the starting patch size for all my modeling, binomial regression modeling, and linear regression modeling. So I'm going to give a little more background before I discuss my results. The logistic regression determines the impact of multiple independent variables to predict the occurrence of an event. Probability is the probability that an event occurs and ranges from 0 to 1. The odds ratio is the probability of success over the probability of failure and ranges from 0 to infinity. And the log odds is the log of the odds. It's difficult to model a variable that has a restricted range from 0 to 1. So log transformation of the odds has a range from negative infinity to positive infinity. To predict the log odds of an event occurring, you can use this equation shown here, where the log odds is modeled as a linear relationship with predictor variables. And then the logistic regression provides the coefficient B, which is the slope, and measures each independent variable's contribution to variation in the dependent variable. A is the intercept, and P is the probability of an event occurring. We didn't use interaction terms, which is the interaction between two or more independent variables, because we didn't think it made biological sense to combine predictor variables, and we didn't think it was appropriate given our sample size. I'm sure you all loved all of that math, and I'll now discuss the most ecologically significant findings for the binomial logistic regression modeling. The backward stepwise binomial selection revealed that the log odds of the coral patch size and surface complexity produced the best fitting model for predicting the probability of coral patch mortality. So here you can see the dependent variable on the y-axis is whether a mortality event occurred, represented as 1 on the y-axis, or did not occur, represented as 0 on the y-axis. And the x-axis is the log of the starting patch size. For reference for you all, there's a table that shows how log of starting patch size values are associated with square centimeters. So for example, a log of -12 is associated with the 0.061 square centimeters, -4 associated with 183 square centimeters, and 0 is associated with 10,000 square centimeters or 1 meter squared. Although log odds, represented by the coefficient b, is modeled as a linear relationship with predictors, when modeling the probability, it's shown as this sigmoidal curve. And here, since I plotted probabilities, not the log odds, this is why it appears curved on the graph, because it's not linear. And the fitted line is the predicted probability of mortality given the log of the starting patch size. As you can see, there's a higher occurrence of mortality at smaller patch sizes, but as the log of patch size increases, there's less occurrence of mortality. So there's a fitted line that's steepest in the middle portion of the patch size data that you can see shaded in red. And this indicates that a change in the log of the starting patch size has a larger effect in this range. Specifically, what we can learn from this data is that corals with a size less than log of -10 square meters or 0.45 square centimeters have a greater than 75% chance of mortality. And you can see this if you look at the left side of the red band. And corals with a size greater than log of -7 or 9 square centimeters have a less than 25% chance of mortality, as you can see on the right side of the red band. So all patches that are situated on the left side of the red band, they're in bad shape, as shown by the skull. And all patches situated on the right side of the red band, they're just stoked, they're thriving, they're chilling because they have a really low chance of mortality. The surface complexity of the reef was negatively associated with the probability of coral mortality, meaning that there are lower probabilities of coral mortality in more complex reefs. And just as a reminder, the surface complexity is the ratio of the 3D to the 2D area of the reef. So a flat reef with no dimensions has a surface complexity value of 1, while a reef with really high topography and many formations would have a greater complexity value of, for example, 3 or 4. So when a reef has a complexity value of 1, the lowest level of complexity, the probability of mortality is 28%. When that complexity of the reef doubles to a value of 2, the probability of mortality drops to 15%. And at a surface complexity value of 3, the probability of mortality drops to around 10%. So more surface complexity is indicating some level of resilience. GENUS, log transformed coral patch size, and surface complexity were the strongest predictors of coral patch growth. Coral patch size was negatively associated with the probability of growth. Here you can see the log of the starting patch size on the x-axis and the probability of growth on the y-axis. So as coral patch size increases, the probability of growth decreases. For example, a coral patch of 0.45 square centimeter has around a 70% chance of growth. Meanwhile, a coral patch with 820 square centimeters or 0.08 square meters has a probability of 60% for growth. Reef complexity was negatively associated with the probability of growth. Here you can see surface complexity on the x-axis and probability of growth on the y-axis. As surface complexity increases, the probability of growth decreases. A reef with a complexity value of 1 has around a 73% probability of growth. And if the complexity value triples, the probability of growth drops to around 60%. Along with these binomial regression models, I also ran linear regression models to understand what variables best predict recruitment rate. And recruitment rate was deemed as the number of coral recruits per 0.8 meter squared circular area or 1 meter diameter circle. And based on AIC values that I mentioned earlier, surface complexity was found to be the strongest predictor of coral recruitment rate. A linear regression model fitting surface complexity on the x-axis and recruitment rate on the y-axis revealed a statistically significant positive relationship of 8.4%, meaning surface complexity explains 8.4% of variability in recruitment rate. And while this is a significant relationship, 8.4 is a pretty low amount of variability. And this just indicates that habitat complexity has some sort of relationship with recruitment, but there are clearly other factors at play. So you'd have to do further investigation to better understand these associations. So after all of these technical graphs and results, what does this all mean? Logistic regression analysis revealed that smaller coral patches were associated with a greater likelihood of mortality, but larger coral patches were associated with a higher likelihood of shrinking. And this is consistent with previous research. For example, smaller coral patches have been found to grow quickly, yet be more susceptible to stressors, while larger coral patches have been shown to be more resistant to mortality, but more susceptible to shrinking. And one great thing about these findings is they help us identify differences in coral characteristics that are either negatively or positively associated with coral resilience. And the better we understand what helps corals survive, the better we can help corals recover from disturbances and help restoration efforts to succeed. The association of coral patch size and coral growth and mortality can be attributed to the interconnected nature of hermatypic corals. Larger coral patches may shrink and smaller coral patches may undergo mortality because larger coral patches have more polyps and they have a greater capacity for energy redistribution towards regrowth, recovery, and maintenance of tissue that has been damaged due to stressors such as predation or disease. But smaller coral patches are more susceptible to full mortality following stress events because there's just less energy available for healing and regrowth. Simply put, larger corals have greater surface area, so they're more likely to succumb to partial mortality and shrinking, but smaller corals, they just undergo full mortality in the face of extreme stressors. And this could also be explained by the life cycle. As coral patches get older, they may become more stable and have greater resilience against disturbances while their growth rates slow down. Competition for space could also influence the low likelihood of growth and high likelihood of shrinking for larger size coral patches. Space on benthic substrate is a limiting resource that affects competition, predation, and stress among coral colonies. A site with larger coral patches and higher coral cover likely has limited available benthic space to grow. And so this could explain the tendency of larger corals to be less likely to grow and more likely to shrink. Results show an important relationship between coral demographic events and reef habitat complexity. Reef complexity was negatively associated with the probability of mortality, with more complex areas associated with less mortality and fragmentation events than less complex reef areas. So why would more complex reefs be associated with lower mortality rates? Reef structural complexity has been positively associated with overall reef health, specifically fish diversity and abundance. Healthy reefs with high fish biodiversity support a greater abundance of herbivores like parrotfish or surgeonfish that graze on the algae and compete with coral for resources and space. In addition to greater biodiversity, highly complex reefs have been shown to have increased resilience to disturbances like wave action and storms. So a complex reef that can withstand disturbances and host a biodiversity of fish may have the ecological resources available to withstand future environmental stressors. So the negative association between reef complexity and mortality can be attributed to the nature of highly complex, biodiverse, and resilient reefs in sustaining higher survivorship, lower fragmentation rates, and lower mortality rates among coral patches. There was a positive correlation between recruitment rate and surface complexity values, with more complex reefs supporting greater recruitment rates. And this further indicates the importance of reef complexity. This could be, again, explained by the nature of healthy, complex reefs in supporting a high abundance of grazers and preventing macroalgae overgrowth. Coral recruitment has been shown to be inhibited by direct contact with macroalgae. So a more complex reef with coral that outcompetes macroalgae for space is more likely to support coral recruitment. And in addition, a complex reef that inhibits grazers that control for algae cover and more benthic space is likely to support higher recruitment rates. As climate change and global warming become more severe, it's evident that conservation of complex reefs that maintain higher recruitment rates and lower mortality rates is essential. And a recent study by Chung et al. In 2021 found that ecological resilience declined rapidly with the loss of habitat complexity, even more so than the loss of corals and herbivores under stressors. And this is because complexity can persist even when corals die. And so this helps the recovery of corals and herbivores. So this further proves that complexity is a vital metric to include when assessing coral reef resilience. Lastly, GENUS was shown to be associated with coral patch growth, shrinking, and fission. In this study, the most dominant genera are Papahānaumokuākea, Acropora, and Montipora. Papahānaumokuākea was positively associated with coral fission events, while Acropora was shown to be negatively associated with coral fission events. Smaller mounding, encrusting, and branching Papahānaumokuākea patches could be more susceptible to fragmentation events, unlike Acropora, which have large tabulate morphology. There is also a negative association between Papahānaumokuākea and coral patch growth, and a positive association between Acropora and coral patch growth. And this could just be due to the nature of tabulate corals to grow quickly and have early reproduction, unlike massive corals like Papahānaumokuākea that are slower growing and have been known to easily undergo partial mortality. So with that, different genera have varying morphological and physical traits that affect variation in responses to their environment in terms of growth and shrinking and fission of coral patches. In conclusion, remember how I said this study uses patches instead of just a subjective determination of coral colonies? By doing this detailed analysis of coral patches, we're finally able to identify specific patch characteristics and site characteristics associated with positive or negative events. And if we had examined reefs in the traditional methods, just relying only on overall coral cover, you would think that there's nothing differentiating the reefs through time. But by using these detailed methods, we identified actual indicators of reef resilience, patch size, and surface complexity. So this means that reefs with greater complexity, composed of larger corals, will be more resilient and less susceptible to mortality events. These findings are essential in informing stakeholders about how to manage and prioritize protection of reefs in the Hawaiian Islands. And what this means is now researchers can just go out in the field and look at simple rugosity and size and use those as indicators for reef resilience. And these tools can equip managers who have either minimal or many resources to better understand reef resilience. Also, this study demonstrates the use of high-resolution 3D structure from motion photogrammetry methods for monitoring and understanding trends in coral reefs. The methods we use in this study can easily be implemented into existing monitoring programs. So even if you can't make these 3D models, by identifying complexity and size of corals in a reef, you can better understand reef resiliency. And anyone can measure these metrics with tools. So this really benefits anyone doing coral reef ecology research. It's critical that agencies like NOAA and other organizations invest in detailed long-term monitoring, because without monitoring, we have no way of predicting, protecting, and conserving these reef ecosystems. Additional time points will reveal future trends in how coral patches respond to environmental stressors, and could even allow for correlation with particular climate or disturbance events. And this study can help develop targeted coral assessments based on these findings about coral patch and site characteristics, such as focusing on small coral patches and low complexity reefs, knowing that they're more vulnerable to mortality events. And I believe if these targeted strategies were implemented, this would significantly improve monitoring programs and protocols in the Hawaiian Islands. So agencies should continue traditional reef monitoring that add new tools and approaches, like those I discussed today, to enhance our understanding of coral reefs. And with that, I'd just like to say mahalo to Dr. John Burns, Dr. Grady Weinberg, and they spent a significant amount of time with me, meeting with me every week in the fall and spring to help me through my analysis and my writing. Dr. Jolene for her support and edits, Noah and Dr. Tom Oliver, Kylie Pascal for her constant support and help the past 2 years, answering all my texts and calls at random hours, and Alex Spangler for helping me throughout this process, starting with the Ikevai summer program, and Danny for all her help tracing coral and checking the data, the Hilo TCBES program and the cohort, thanks for all your support. And of course, all my friends and family for the endless support. I appreciate you all so much. And lastly, thanks to the Hilo Ikevai students. Thank you all, because without you, I could not have done this. And here's just some pictures of those who helped me along the way. I want to particularly point out the Zoom photo with me, John, and Grady, because this is basically how I spent the past year and a half on Zoom with these advisors, with me looking very confused while John and Grady helped explain stats and coding to me. And then another Zoom picture of all the Ikevai students and my family, my Hilo crew, Oahu crew, neighbors. Thank you. And I will now open it up to questions.